Seconds. So as you have seen in the previous presentations, maybe I have a couple of housekeeping items, then we'll do the poll, and then it will be up to you.
Okay. Great, thanks.
Hello, everyone. Thank you for joining us for this last presentation in our preclinical track, in the preclinical session, where Lara Clemens will talk to us about the BIOLOGXsym to demonstrate hepatotoxic potential of Tocilizumab. Before we get started, a couple of housekeeping items. We take your privacy rights seriously by attending this event or participating in the Q&A session. You are allowing us to contact you for follow-up. You may ask questions via the Q&A tab on your dashboard at any time during the presentation. If you need technical assistance, please use the chat tab. To begin with, let's start with a quick poll just to get an idea about how many of you are involved in the development of the biologics. If you could just quickly answer on your knowledge on your company in the development of biologics. We'll wait another 10 seconds. Okay, and hopefully everybody had the chance to cast your votes. Interesting. We have about 50% of the responders said that their company is not involved in the development of biologics, so have a chance to learn something new. And the rest is split about evenly between yes and unsure. So go ahead, Lara.
Great, thank you so much. Great, thanks for the introduction. To start, I would like to make sure to acknowledge all of my colleagues, both with DILIsym Services and the University of Pittsburgh, all of whom have put a lot of effort into this project. There have been a lot of moving pieces and a lot of components that I will talk about today, and that couldn't have happened without them. And I would also like to note now that a version of this work will also be presented at the Society of Toxicology in March. Okay, so getting started, biologics can address many unmet clinical needs. However, biologics-induced liver injury, or BILI, can slow or halt therapeutic development. And so to evaluate new biologics for BILI liability, we developed BIOLOGXsym, which is a new quantitative systems toxicology software. For an exemplar compound, we started with developing a model of Tocilizumab in BIOLOGXsym. So Tocilizumab is an interleukin or IL-6 receptor antagonist monoclonal antibody. And it is commonly used to treat inflammatory diseases such as rheumatoid arthritis. Notably, signs of liver injury, such as elevations in the liver injury biomarker ALT, are seen in about a third of patients administered Tocilizumab. And in about a third of patients, this is lower level elevations of ALT, but the more extreme cases, as we've heard about being signs of more severe liver injury, are seen in 1% to 2% of patients. Okay, so our goal here is to develop a model of Tocilizumab to predict and hopefully gain insights about its potential for hepatotoxicity. And so this involves many moving pieces, and I will try and zoom in here. So first, we use information about the exposure to predict what concentration of Tocilizumab will infiltrate the liver. We also incorporate mechanistic models of relevant liver biochemistry and toxicity mechanisms, including both on and off-target effects of Tocilizumab specifically. And lastly, we also use experimental information about the potential BILI mechanisms of Tocilizumab to inform and parameterize our model. Importantly, these BILI mechanisms are studied by our collaborators at the Drug Discovery Institute at University of Pittsburgh. They developed a human vascularized liver acinus microphysiology system, which I will refer to in the future as VLAMPS, to look at how biologics impact cellular function in a liver-like environment. So for the sake of time, I won't go into that in great detail. They will be presenting their work on their system and some of the results from this project at the Society of Toxicology in March. But using their system, they evaluated how tocilizumab, with or without additional IL-6, affects key liver responses, including buildup of reactive oxygen species, steatosis, and changes in CYP activity, all of which we were able to use as inputs into our model to inform and parameterize our model to then predict hepatotoxicity. And that's this center part here, is taking all of these components and using that to predict BILI. Okay, some of our results. So just briefly, I will talk about some of the exposure and the BILI mechanism components of the methods that I just introduced. Specifically, we developed a PBPK model of tocilizumab using the BIOLOGXsym module in GastroPlus, and we fit this to available clinical plasma data, and then use this to predict the concentration of tocilizumab in the liver interstitium as one of our inputs to our model. Specifically, we did this for an 8 milligram per kilogram intravenous administration of tocilizumab using a 4-week, once every 4-week dosing for 12 weeks. So our simulations will be 12 weeks, and the 8 milligram per kilogram dosing is a common clinical dose. Okay, additionally, and again, I won't spend too much time here on the experimental output. If you're interested, again, this data will be presented in March. But we do have very valuable outputs from the VLAMP system that looks at what happens if you add tocilizumab with or without IL-6 or IL-6 by itself to a liver-like environment, and how does that affect key outputs, such as in this case, I've included the data for CYP3A4 activity, but we also have data reflecting ROS buildup and steatosis, and we use this data to parameterize our model. And of note here is that if you add IL-6, then you have a decrease in CYP3A4 activity, but if you add tocilizumab, you recover that activity. Okay, getting into the mechanistic model that we built for tocilizumab within BIOLOGXsym, we include many of the main pathways that will affect the liver. Specifically, we include on-target effects and off-target effects. I mentioned at the beginning that tocilizumab is an IL-6 receptor antagonist. So tocilizumab can bind to the IL-6 receptor. This takes two forms. There's a membrane-bound and a soluble. We include both of these signaling pathways in our model. We implicitly represent membrane-bound IL-6 receptor and have tocilizumab modulate and create an effective IL-6 concentration that would mimic the signaling seen from a membrane-bound receptor. But we also include an explicit representation of IL-6 or tocilizumab binding to soluble IL-6 receptor. And then we include key impacts of the IL-6 signaling pathways on hepatocytes. And for this, we've included some, including changes in CYP activity, and changes in hepatocyte regeneration or hepatocyte turnover, and additionally macrophage recruitment. We also have key outputs of, for instance, C-reactive protein. Oops, apologies. All right, and then in addition to the direct on-target effects, or the where again here on target is specifically those involving the IL-6 signaling pathway, we also included off-target effects of additional effects that tocilizumab may have that do not necessarily involve the IL-6 pathway. And so here we also include a buildup of reactive oxygen species and changes that may lead to steatosis. Okay. However, our original goal was to see whether we could predict the hepatotoxic potential of tocilizumab using this model. And so what simulation to answer this question? So first of all, we set up 4 simulated individuals, and we varied some of the baseline characteristics of these individuals. We varied things like body weight and different mediator levels, but importantly, we also varied some of the relevant BILI mechanisms. So for instance, susceptibility to ROS toxicity. And so we developed 4 simulated individuals that we will use for these simulations. And in each case, we also started them with elevated IL-6 levels compared to a normal healthy human. And this was to mimic an inflammatory disease state, much like patients that would receive tocilizumab. To each of these simulated individuals, we gave them each a dose of 8 milligrams per kilogram intravenous tocilizumab once every 4 weeks for 12 weeks. And using this simulation, we were able to look at how tocilizumab would affect those patients. But in addition, we were how tocilizumab-driven change in activity could impact the toxicity during drug co-administration. And so to do this, we simulated co-administration with a gram given 4 times daily, as acetaminophen is metabolized via CYPs. And that is one example that we can use to look at this type of dynamic. Okay, starting there, we considered ALT level, the peak plasma ALT level, as our main marker of liver injury. So in this plot down on the bottom right, we have different administrations, and I will start with just the. And for each of the 4 individuals, we have 4 different dots for each administration, and we demonstrate the peak at those 12 weeks for each individual. And then on the plot, we also have marked the upper limit of normal for, and so we have at the bottom times the upper limit of normal, 3 times the upper limit of normal, and 5 times the upper limit of normal. And threefold is considered pretty significant ALT elevations, but about a third of tocilizumab patients also were seen above 1-fold. For acetaminophen by itself, for our 4 simulated individuals, we see that 2 of the 4 individuals have significantly elevated ALT. For tocilizumab by itself, we can see that all of our individuals have some amount of ALT elevation, and one of our individuals has particularly elevated ALT. When we combine the 2, when we administer both acetaminophen and tocilizumab, we can see a significant in most of our patients. And notably, 2 of our patients have very high ALT compared to each of the individuals. Okay. One of the other questions that we were interested in is how do the different effects of tocilizumab, specifically the on-target pathways and the off-target pathways, affect this response? Of how much of this is driven based on the changes in CYP activity and hepatocyte regeneration compared to, for instance, the off-target only effects where we would simulate only the additional impact of tocilizumab-induced ROS or steatosis. And so we broke this down into both of those simulations where we represent only the on-target effects, only the CYP activities, hepatocyte regeneration, additional macrophage recruitment. And what we see there is we still do have significant ALT elevations in 2 of our individuals. And when we instead only simulate the off-target effects and leave the changes in CYP activity and hepatocyte regeneration out of our simulation, then we also still have significant elevations in ALT. And so this demonstrates that both of these are contributing to the elevations that we're seeing here. Okay, so to summarize, we have developed a new software to predict BILI for new biologics, and this is called BIOLOGXsym. And using this, we modeled on and off target effects of tocilizumab and found that the hepatotoxicity that we predict for tocilizumab is consistent with the clinical results that are seen. And additionally, we've been able to use BIOLOGXsym to investigate how co-administration of tocilizumab with acetaminophen can lead to more significant ALT elevations, and how the on and off target effects of tocilizumab will impact those the hepatotoxicity seen there. And so This demonstrates that our QST model, BIOLOGXsym, has the ability to predict potential hepatotoxicity of biologics. And importantly, these methods are generalizable. We can take the approach that we used for Tocilizumab and apply it to other new biologics. And that offers a really nice generalizable approach to rapidly evaluate liver safety in new biologics. And hopefully we can also gain insights into the main mechanisms of toxicity and potential drug-drug interactions. All right, and again, I would like to thank all of our collaborators. This was work funded by the NIH. We had a phase 1 SBIR grant, joint lead for DILIsym Services and University of Pittsburgh. All right, thank you very much. I'll take questions.
Okay, thank you, Lara, for the great presentation. Unfortunately, that's about all the time that we have. We are actually running a little bit over, so I'll have to direct everybody who has any questions to try to contact you directly. I'm sure you'll be happy to discuss that offline. So thanks everyone for joining us. This webinar has been recorded for playback and will be available on our website and the YouTube channel. This concludes our session. Please head over to the lobby for the next presentation. Actually, the next event in this track, we have the panel discussion. So we'll look forward to seeing you there.